Morning, everybody. Andy DeHennis, here to talk about some of the lessons learned along the translational highway. And as Tim mentioned, just part one here, the foundations of disruptive technology and scientific challenges. I'm with Senseonics Incorporated. We're developing a continuous glucose monitoring device. And I'm going to talk a little bit about that and some of the things we've learned along the way for the past few years as I've been there. So yeah, as I mentioned, it's just some case examples I'm going to refer to. This is a photograph of our device. And I'm also going to refer to the artificial pancreas a little bit, just because I think it's a good case study to talk about the translation of technology. Okay. Oh, is this not a microphone? It's just for recording? Okay. All right. Yes, so Senseonics CGM system. So first is a smart sensor. Our sensor is actually subcutaneously implanted just under the skin in the upper arm. This is a photograph here next to an M&M, which is just kind of an icon that people can recognize. It's fluorescent based. It lasts for 3 months. It has an equilibrium recognition mechanism on the outside of the sensor. So the transducer itself is not consumed by binding to glucose. It uses NFC communication and wireless powering, and has no battery. So we have an implant as part of our system. We also have a wearable as part of our system. So this transmitter powers the device. It communicates with it, takes a measurement to enable the continuous monitoring. Also calculates and stores the glucose data. So alerts and alarms are on body through a vibration motor. It's rechargeable, and it also has Bluetooth communication that talks directly to your smartphone. So this Bluetooth communication enables displays of alerts and alarms, as well as calibration entry. And as mentioned here, this is an investigational device. So we're still in various stages of approval, doing our research. So really what I want to talk about here is kind of the original technology. So the original technology was the concept to have an indicator that can bind glucose and change from a low level of fluorescence to a high level of fluorescence. And that's basically the idea and the concept and the invention originally. That was many years ago and has transformed into essentially what the users experience, which has very little to do with the invention itself. It's almost transparent. So in the end, the system and the concept turn into a wearable that's body worn, attached with an adhesive, and an app. So this essentially is the translation, right? Turning it into an actual product that people with diabetes can use to help manage their disease. Going into a little detail about how the device works. So it's a fluorescent base, so it has an excitation source. We use that as part of our referencing system. And then the integration of that fluorescence is what is dependent on glucose and gets transduced and communicated by the sensor to the transmitter and then out to the smartphone. So really to talk about the ideal translational process. So having been to graduate school myself, doing a thesis, really I thought that, you have this great idea, this invention. What can you do with it? Oh, well, you can productize it, turn it into a product, take that product, do some clinical testing, and then do a regulatory submission. And that's development. Turns out that that's kind of not the case, I guess, in practical implementation. So really the reality of it is the overlap between all of those stages, right? So you have this idea and invention, you start to productize it, turn it into a form factor that's usable. Sometimes you need to go back. Sometimes you need to revisit that original concept and do modification, tweaks, optimization of that concept to figure out how to turn that into a usable device. Taking that then usable device, starting to do some clinical testing, right? Understanding the impact by the physiology, the immune system, with either the form factor or the concept itself. And then sometimes need to go back and revisit the productization, whether it's the encasement materials, the packaging, the interface, to optimize it even further. And then potentially a regulatory submission. So slightly different than at least what I had originally envisioned, but there's a lot of benefits in understanding this and understanding that you need to translate to these different stages, and taking that and doing some of that as early as possible in situations where you can. So really, just to go into detail about these 4 categories, I have some bullets here that talk to each of them, right? So really the initial concept, flashy, exciting, can potentially claim a lot of different things on that. You have to be careful with that, about what you claim about the original concept, because when it comes to the practical implementation of it, it may end up being slightly different. So set expectations in somewhat of a reality is part of what we've seen. So sometimes the idea of being so different from anything out there isn't the best thing either, right? So if it's so different that nobody can understand it, then they have difficulties, whether it's clinical testing or regulatory approach. You have to be careful with that as well. So if there's something you can grab onto that's been previously developed, or even doing your idea in stages can have potentially benefit as well. Not to do too much critiquing here, I guess, or say anything that has any blasphemy behind it, but it's not necessarily the best idea to do an integrated circuit from the get-go. Saying that a biocast maybe isn't the best idea, but it's the reality of what we've seen, right? So the faster you can find a prototype to get some kind of clinical testing will enable that ASIC or that circuit to be all the better, right? So you can understand the dynamic range or the levels of signals that you're going to see when you really get in vivo, as opposed to kind of doing the calculations and the bench level testing. So really something that can enable you to test in vivo has a lot of value. And this is, a prototype that we did with SMDs from DigiKey, dye from SemiDice. We put it together and it enables a lot of insight just going through the testing. So utilize early testing as much as you can. Understand the biofouling and the bioreaction from your device. Whether it's in vivo or ex vivo or it's a wearable, that interface is usually part of the crux of the development. And for us, this is actually a picture of the fibrotic capsule that develops from our device, shown here. And we've been doing clinical studies for a couple of years now, and understanding what's going on in vivo with this fibrotic capsule formation, with the immune system that reacts to your device. Having that studied early on has had a lot of benefit, and understanding how it compares to other sensors. So this is a glucose oxidase sensor and the fibrotic capsule that forms around it. And for our device, I would say that the last couple of years of in vivo testing, we have a lot of experience, and there's a lot of value to that experience in understanding that immune response. Because essentially we're studying the immune response inside the body with a flashlight. So we've seen a lot what goes on, and there's a lot of lessons learned from seeing some of that. This is a slide really just to talk to intellectual property, right? So everybody's, patents kind of either filed them or talked about them. This is essentially our patent landscape, where we've kind of covered every corner of the device, right? The implant, the wearable, the communication, the user interface. And you have to make sure that not only your original idea is covered, but anything that may block you as you make the final device also has to be addressed. And the one other thing, and this is something to be mindful more recently. So I think in 2013, the Patent Office changed their policy. I don't know how many are familiar with that, but now it's got a first to file, right? So if you have an idea and you put in your lab notebook, it's not really an idea yet. It's only an idea once the Patent Office gets it. So it's something to keep in mind. Then at some point, this is where we did our ASIC, right? So after we understood some of the in vivo dynamics, we did a full ASIC where we could really shrink the component count, make it a scalable and manufacturable device, and start to move forward with our clinical testing. The other thing that we found is, although as an engineer you think you can understand the problems of the world, or at least I did, there's a lot of benefit in just talking to people. Do a focus group for people that are going to be treated with the device that you're developing. This isn't ours. I don't know if I stole this off the web. But talking to people, and one of the things we learned is how important that wearable component is. So that's something we were like, oh, make the implant smaller, and we were really fascinated with the scalability of that. But the people with diabetes were really almost self-conscious about the size of the wearable, right? And how important that played an impact in our development of their device. Really, another part of this is to polish and test and enable a testable platform. So know that somebody is going to be walking around with your device in a clinical study or as a product, and you're going to have to provide information to them that's understandable, right? And sometimes it's information to them when the device is working, and sometimes it's information to them in kind of corner case error situations, especially early on in development. So spend some time thinking about those situations where somebody's going to just sit there looking at it like, well, what do I do with that? And then they're going to call, and then they're going to expect you to tell them what to do anyway. So doing it early on has a lot of value as well. This is actually a slide showing the artificial pancreas work from UVA, the Center for Diabetes Technology. So really this kind of represents the different features that a user with an artificial pancreas expects. Signals of hypo, signals of their glucose, signals of their insulin pump, and signals of hypo, all on a kind of streamlined interface. So thinking about how you can simplify it, both for enabling the communication as well as simplifying so that they are compliant with the device, can have a lot of benefits. They actually went so far as to put it on a screen inside Google Glass, and one of the guys took this picture. But making it easier on the users has a lot of benefit for compliance. Really, in speaking about compliance, however you may enable it, it may not happen, right? So you also have to build in features to your device and system that can teach you about what they might really be doing, as opposed to what they say they're doing. And one of the features we did in our devices, we built in an accelerometer. We can understand when they're sleeping or awake, whether they've dropped it, things like that, because the sensor kind of never lies, as opposed to what may happen with the device out in the field. You just kind of, it's out there, and you got to trust that the transduction will enable a little more information than other sources. So the other thing to think about is once you have this device and you've been developing it, is how can you clinically prove it that it has benefits, clinically prove its accuracy. So a lot of the different technologies that you develop, at some point you have to say, okay, there's some justification for this. And in diabetes, the way that's done is actually to go in clinic, take a reference standard, monitor it every 15 minutes, and then essentially calculate your accuracy to that standard. Right? So you have to do various points and do statistical powering to enable the justification of your device, really. Any questions along the way, feel free to ask. So just as I'm going through here, go ahead.
I was curious how much time occurred in your case from the initial publication of basic research to where you are now?
So the publication of the basic research, so we actually started 17 years ago. It was a oxygen sensor company. Using the same basic fluorescent principle, LED, photodiode, and that morphed probably in the early 2000s into really looking into glucose and developing a sensor that would transduce glucose. That's when we kind of came out with that molecule I showed earlier. So that was in about the 2003 timeframe. About 2007 or 8, right in that timeframe, is when we started doing human studies. Very eye-opening in terms of what happens with the biological response, and a lot of insight was gained by that. And then the last 5 years or so has really been going through the clinical trials, both in different parts of the world, as well as we just finished our a pivotal study in Europe just this summer. So a good number of years there, yeah.
So how much did you spend on your IP portfolio?
Oh.
US.
Line item on our budget in the past decade or so. Yeah, millions of dollars. Yeah.
So you have to do it through the funding you raise.
Yes.
Kind of come out of research grants.
We're 100% VC funded. Yes, very patient investors, understanding the technological approach we've taken, as well as the potential market and solution that we're working towards. So.
How's your experience for the FDA process?
Wow, that's a good one.
They're FDA.
Yeah, we're not FDA approved yet. It's been informative. That's all. Yeah.
I can take it.
Yeah. Okay.
In your productization, how much did you analyze prior devices similar in form factor, I would say, for example, the Bio, in terms of migration, in terms of packaging, that type of thing?
We studied.
Resembles a Bio.
Yeah, it does. It has some similarities to Bion. We don't have a battery, so it's a little smaller than the Bion, the way it was formed. I've seen the Bion. It's a little bit longer.
Cylindrical migration is a...
We insert it just under the skin, right? And we don't see that much migration when we do that, as opposed to deeper in the body.
Cracking of the packaging, I don't know what type of packaging you are using.
It's polymer based, and the cracking has been very minimal. It's a continuous, so it doesn't have any air to crack, so it's able to...
In 90 days, you don't see ingress of leakage fluid inside it.
We've had to build a package that's robust to handle some of that, yes. So I got 2 more slides. Let me get 2 more minutes here. Tim gave me the 2-minute duty. So this is another way of proving it in terms of its performance. This is a Clark error grid where you talk about having accuracy in the A region here clinically, so you're giving people the right information, and the E regions here having essentially misleading information on your screen. You got to be very mindful of that in terms of the standards that are out there for the specific devices in the different industries. So I had one last slide here, and then we can do some more questions. Really speaking to the FDA or regulatory process. But in the end, it's documentation, right? So what you submit is documents. They don't necessarily want you to send them a demo or anything like that of here's my device, check it out, play with it. They want to see that everything's sound, both in the design, the development of the system, as well in the proof of it clinically and its performance, right? So documentation is a part of medical device development. And really to end on part of that and kind of create excitement about some of that documentation is there's a lot of benefit to it, right? So one of the benefits of doing that documentation and taking it so far that people are then using your device is you get to see the benefits that the people have when they use it. Right, so this is actually a paper that we published about 9 months ago or so, where somebody in an unblinded study is using our device, right? They go to sleep, so our accelerometer there, you can see the activity level, so we have high and low, and essentially they went to bed. The low activity is just a sign that they're really sleeping. Our device then alarms and alerts because they're going hypoglycemic. Right? You can see that they then get up and remediate that situation. And then sometime thereafter, after they've recovered from the hypo event, all really traced by the SMBG confirmation points that they have to take, especially in the study with a not approved device, you see them go back to bed. So this is the benefit of that documentation, right? So I heard Professor Liang talk about 60601. It's worth slugging through that at some level and understanding what you have to, get to then have somebody use your device. It's pretty cool. Questions? Yeah?
So I just wanted to ask, do you think that the 20-year patent coverage is long enough then for medical devices? Should it be longer in that space?
Well, in some of the patents, you can do a continuation in part as you add different ideas or parts of the idea to that and enable some longevity to that. So it depends. Yeah, we're at the expiration of some of our original core patents, but some of our newer ideas have added on to that and probably provide enough protection to enable protection of our ideas. But also having the freedom to operate from those patents on the books has a lot of benefit as well.
I used to work with some people who
started this company, GlucoWatch.
Yeah.
Familiar with them, Janet Tamata and Brad Phipps. And I know one of the challenges they ran into was that the interstitial glucose measurements don't necessarily track the blood glucose, which is what all of the kind of recommendations are based on and guidelines are based on. Is that something that you need to, so from the translational aspect, that seems like a major kind of scientific challenge and something that you want to look into, but are you the one to look into it, or is there enough kind of literature out there where that's not an issue for FDA moving forward?
So that's a good question. I'm familiar with the glucose watch work. And as we had a watch early on, somebody said, is that the glucose watch? When it wasn't, it was just a watch to read the implantable sensor. Yes, there's some truth to that in terms of the physiology, right? And there's what we call kind of as lag time, right? There's a lag time about 5 to 10 minutes. So what you have to do in the development of a CGM is learn how to compensate for that in your algorithm. So there's algorithmic approaches to that can enable higher point accuracy to give subjects what they need to manage their diabetes. So it is a part of it, but it's not critical enough so that it doesn't enable a lot of insight. And one of the benefits of CGM is trend, right? So on your screen, you not only have the point, but you also have the trend. And that's one of the add-ons that's not in SMBG, right? SMBG, you see you're at 100, you think you're okay. But if you're 100 going down, that's a very different story than 100 going up. And so it's the rate arrow that also provides a lot of benefit.
I guess the interesting question would be as skin temperature changes and you start affecting basically pre-capillary, post-capillary pressures and you change.
Yeah.
Right?
So we actually, we've seen,
I'm actually going to hold just to keep
sorry.
time. So because we want to keep someone on schedule. Part of the structure is, Andy will be around at least for the break.
I'll be able till 10, yes.
Yeah, and so we'll have time to break. And then also we're going to have a panel at the end, or we used up one speaker spot to actually go deep into some questions. And actually there could be multiple questions you want to hear from all of us. So let's thank Andy at this time.
Okay, thanks.